Thank you so much, the organizers, for inviting me to moderate this next session. The whole day is just a fabulous group of talks, and I'm excited about this next session. The title of this session is as follows: Adaptive versus Innate Immunity in Alpha Beta T cell / CD19 B cell Depleted Haploidentical Transplant. Which one matters? We have two wonderful speakers during this next session. The first speaker will be Jürgen Kuball, who will give us a talk with the title, The Biology of Gamma Delta T Cells. Is this the secret for the success? And the second will be Atg Bertaina, whose talk will be entitled Alpha Beta T Cell CD19 B Cell Depleted Haploid Transplant, a platform for immunotherapy. I'm really excited about these talks, and we will turn it over to Dr. Kuball, who will give the first talk of this session. Thank you.
Thank you very much for this very nice invitation. Here you will find my relevant disclosures. Today I would like to talk about ex vivo T cell depletion, a platform for immunotherapy, and to start with what I think is a very important, completely underestimated aspect, that allotransplantation is nothing more than a game of diversity and numbers. And if you look at diversity, just looking at the T cell compartment we are infusing, of course, we have a lot of alpha beta T cells, which is the main thing people think is important. But of course, there are also gamma delta T cells, T regulatory T cells, and NK T cells. And balancing these cells is important if we infuse a graft, but also later if we infuse a DLI. And I can only recommend a review we just wrote, you can see here on the right side. If you simply analyze T cell numbers infused into patients during a stem cell transplantation with a graft and analyze what is the outcome of these transplantations, then apparently it looks like that we count, but we don't care about numbers, because with a certain threshold exceeding for T cells, apparently you buy in the only toxicity in terms of graft versus host disease, but not graft versus linear control. So I think it would be wise if we reconsider the number of cells we are infusing into a patient and have a certain limits for certain types of transplantations, which of course differ between haplotransplantation and unrelated donor transplantations, because mismatches also differ. This means less is more, and less is more is also something we should consider if we look at how can we restrict diversity in numbers, either with, for example, smart Atg dosing and also graft engineering. If we first start with Atg, we know that Atg is frequently used at different dosages at different centers, also with different timings, which is meant to facilitate engraftment and reduces GVHD because it depletes host T cells and APCs. On the other hand, it's also doing a graft T cell depletion, in particular if it's giving very shortly before we infuse the stem cell transplantation product. It reduces GVHD, but can also increase relapse rate and increase infections. And just to show if we don't have any restrictions on Atg in terms of dosing in the patient, what can happen is that apparently there are some patients who have by chance an optimal Atg dosing and others who don't. And this has, in terms of overall survival and event-free survival, really a difference between 30% and 70%. And this is heavily impacted by the circulating lymphocytes who are binding Atg at the moment once we infuse them. And that's the reason as to why we propose to not only dose Atg as we currently do, but in particular also look at the circulating lymphocyte counts, which tell us how much Atg we need to give to a patient. In particular with patients who have lymphopenia, it is recommended to reduce the dose of Atg. Another way is to restrict upfront through graft engineering the cells we are infusing into a patient. And again, showing the different options, what we can infuse into a patient. And the question is, of course, what's the most important one? And this is a paper that has been published a couple of years ago, where people in an unbiased analysis looked at what are the cells we find in the tumor. And this includes also hematological malignancies like lymphoma and multiple myeloma, and what associates with a positive outcome. And number one are gamma delta T cells in these ones, and of course alpha beta T cells. Not saying that gamma delta T cells are the most important one, and that NK cells are the worst one, but that there are multiple layers of cancer immune surveillance which we need really to understand, and gamma delta T cell represent for sure one important layer. Control by graft engineering started with CD34 selection and then evolved to CD3, CD19 depletion, and then went to alpha beta T cell CD19 depletion, which is also very important to understand if you look at, for example, also most recently published randomized trials where you randomize, for example, CD4 selection with a certain ATG regimen to other regimens, because the way how you do this might heavily impact the clinical outcome. Just an example of our cohorts where you see that alpha beta T cell depletion is very efficient in these cohorts. We didn't do B cell depletions, what we in the current protocol are doing. And yes, most importantly, in the time of pandemia, yes, you can freeze cells and you can thaw them again, and they engraft very well in the patient. This is a summary we collected of published trials so far, and just referring to our own trial, which we used for related and unrelated donors, where you see that acute GVHD is of course still present, but in particular, and I think that's the most important outcome, chronic GVHD is heavily reduced in terms of extended chronic GVHD, which really makes a huge quality of life difference. Furthermore, acute GVHD mainly occurred after we started infusing DLI, so meaning you have a very early also window of opportunity for immune interventions. The major goals of change strategies, and this includes also post-allo size, of course, reducing acute and chronic toxicity, adding early on additional immune interventions, which is clearly possible with this platform, and allowing a better elevation of add-on medicine maintenance strategies, I think, which is in our perception one of the main issues also for the next 5 years, better understanding which drugs we need to give for how long to remain remission. Limitations are that apparently, if we look at studies who are investigating how the immune repertoire is coming back, these studies are very fragmented, have different baselines, have different timings and looking at this. So I think this is really for the community important to more standardize also these moments of assessment after transplantation, because at the end of the day, that is what is helping a patient to control leukemia and control infections, how exactly the immune system is coming back. And this means also that this differs between the different platforms. So for example, if you have ATG, of course, it's mainly an alpha beta T cell depletion. If you have post-allo-Cy, it is partially an alpha beta T cell depletion, but I think also dysfunction immune cell repertoires. And of course, if you have an alpha beta CD19 depletion, we maintain NK and gamma delta repertoires. So meaning also depending on the drug you're using later, this has an impact. If, for example, we take checkpoint inhibitors, they have a high risk after ATG depletion to still induce a GVHD, while the risk is lower with post-allo-Cy and also with alpha beta T cell depletion. However, there's also a role of chemotherapy in immunological diversity, which we discovered during the last years, where we saw that if despite a very controlled alpha beta T cell depletion, we have huge variations still between different patients in how the immune cells are coming back, as you can see here. We further analyzed potential factors, and we came down to the fact that apparently these patients had different fludarabine levels, where you have an optimized dose, where you have an optimal immune reconstitutions, and either patients are too high and too low, in particular for adults at higher age. You have, depending on the kidney function, a worse fludarabine targeting level. And this is not heavily captured or well captured by the current guidance how to deal with fludarabine. So that's the reason as to why we started a randomized trial, which is now close to recruiting. So hope we have first data mid of next year, where we use the alpha beta T cell depletion platform with ATG early, where we know that this is not running over the graft, so we don't further look at the dosage here with busulfan area under the curve, and the only difference we allow is fludarabine, either as we've dosed it usually or individualized. And then after that, an alpha beta T cell depletion. So looking forward to these data, and bringing us hopefully to a platform where after transplantation, we can do immune intervention. I want to at least discuss 3 or 4 different options. The DLI is, of course, the first one. I just want to remind everybody that in the early post-transplantation phase, of course, we have a lot of inflammation, depending on the platform and our platform, gamma deltas and NK cells, but still, you should avoid too much DLI too early, and that's what we try to capture here again, that of course you can give a DLI in the T-cell depleted graft rather early at 3 months later, but the dosage should be lower. And of course you can increase over time the dose in these patients, I think, which is a very important one also to control a leukemic relapse. And this is just one of the examples where we guide then DLI depending on the MRD we are measuring, as you can see here, very successfully. And last but not least, I would like to jump, of course, in the 3, I think, major interventions everybody is now interested in, which you could also use post-allo transplant. The one is CAR-T, the other one alpha-beta TCR engineered cells, and of course, from our side, gamma-delta TCR engineered immune cells. And I don't want to tell too much about CAR-T, because I think there are a lot of data out there. I think in haplo setting, not so much, but the general feeling is that graft-versus-host disease risk is rather low. And I just want to jump on this one a little bit more, where we do alpha-beta TCR engineering, because also this is really upcoming, but has a lot of hurdles because, of course, of partially mismatching TCRs, endogenous TCR expression. And as I talked of alpha-beta TCR depletion or T-cell depletion, for the allo transplant, we use the very same antibody to entertain the idea whether this is also useful to enrich alpha-beta TCR engineered immune cells. And why? Because this would be very elegant, because then we have already a GMP grade ingredient which we can use for engineering. And what we did is we used T-cell receptors, which are partially murrinized, coming from Wolfgang Uckert from Berlin, where we assessed whether an antibody, which is directed either anti-alpha-beta TCR, the GMP grade, against which domain. And as you can see here, we mainly mapped it to the beta chain, and we also identified certain amino acids which are important that the antibody is binding. So meaning if you would theoretically then mutate these binding sites, in this case to murine amino acids, because we showed that this antibody is not binding to a murine alpha-beta TCR, you could avoid binding and maybe use it for enrichment of cells. And this is what you see here. So by simply taking a human alpha-beta TCR, which you use, for example, to redirect against WT1 or New York ESA1 in this case, and you introduce just 4 point mutations into the constant domain, you will be able, with this GMP grade alpha-beta TCR antibody, to highly enrich your engineered immune cells. So I think something I can really invite everybody to test also with his own TCRs, because it really has an advantage of getting purer and better populations. At the very same moment, we thought, is it also possible, because then, of course, we have mutated sites, also to engineer another antibody who's recognizing these mutated sites, and which we can use then later for depletion strategies in vivo in case of toxicity, because a lot of these T-cell receptors also cross-recognize healthy tissues. And this was a little bit more complicated, but we still saw that we can generate such an antibody, which we humanized, based on an existing anti-murine alpha-beta T-cell receptor binding antibody. And this is not perfect. It works quite well, as you see in the next slide, where in particular, and we don't understand exactly as to why, a certain stretch of 9 amino acids is the best one for binding and allows a partial depletion of engineered immune cells. But again, this part of the work needs, I think, further modulating of this interface. So to summarize these data, on the one hand, we have already a GMP-ready interface where we can use the currently available anti-alpha-beta T-cell receptor binding antibody to deplete non-engineered immune cells if you introduce these point mutations into your T-cell receptor. And we have another antibody generated in our lab, which can be used to deplete again then these engineered immune cells. But the second part needs, I think, more engineering strategies. Bringing us to our last topic, the gamma delta T cell engineered immune cell, the TECs. And I think I talked in the last years a lot about different gamma delta T cell receptors, in particular about gamma 9 delta 2 T cell receptors. So just to summarize these data, we have shown that an important molecule for recognition is BTN3A1, and that this is modulated by Rho-B. Another group showed that BTN2A1 is directly binding to the gamma delta T cell receptor, that the gamma chain is certain parts are very important, and that potentially maybe CD45 might be an interacting partner for BTN3A1, but that's still an open game. Now, we showed that the TCL receptor affinity of this receptor is very important in interaction, and that there is a certain turnover of BTN3A1 which modulates this recognition mechanism. So at the first sight, rather complex, but makes a lot of sense because these are all self-proteins which sense metabolic changes early during cancer development. Most recent paper also again emphasizing how these molecules are working together. Totally underestimated so far is still that there's a huge diversity in the gamma 9 delta 2 T cell repertoire, even also after other transplantations. So for example, here just from a healthy individual in blue, you see a very potent gamma delta T cell clone, which has a high frequency, but you see also a high frequency clone, which is a very poor in activity. So meaning that not all gamma delta T cells you see in the peripheral blood might be active, makes it very difficult to make from polyclonal gamma delta T cell drugs, but also challenging to understand really after transplantation which gamma delta T cell clone is truly active and helpful. To overcome this, we said just let's take an alpha beta T cell as a carrier and introduce a gamma 9 delta 2 T cell receptor, because you uncouple the innate-like receptors from education and tolerance. And furthermore, this particular receptor, we used reprogram CD4 and CD8 positive T cells. You generate help then for these engineered immune cells. And we brought this concept into the clinic. Also here, a summary of the papers where we first described the concept, then defined in the lead structure we are now using in the clinical trial. We described the mode of action, looked at efficacy and safety in different models, and of course set up a GMP-grade production process to be able to recruit now in the second dose level patients into this clinical study. However, I think the new way and game to go is looking at the delta 2 negative gamma delta T cell receptors, and there still is a lot, I think, to discover. I showed you that high frequencies associate with a positive clinical outcome. We punched out gamma delta T cells together with Paul Fish, really also a lot of credits to him, from breast cancer patients, and we showed that these receptors can be active against breast cancer cell lines. Though there's a lot of controversy, another group states that the T cell receptor, gamma delta T cell receptor, is not so important, but more NK cell receptors are important for this tumor recognition. Regardless of the mode of action, we tumbled during gamma delta T cell receptor specificity recognition over quite an interesting one, because usually people think these receptors should not recognize classical HLAs and should not be able to mediate alloreactivity. However, we described a couple of years ago now a gamma delta T cell receptor which senses HLA-24 at the cell membrane selectively on tumor cells, and this mechanism does not depend on a specific peptide. But we think it's rather clustering how the HLA is at the cell membrane of a tumor cell, which simply differs from healthy cells. And this could be a very useful, because you basically could, for example, Japanese population, treat cancer patients who are HLA-24 positive. Is this true? Also in mice, we got NSG-A24 mice from our collaborators in Japan, and you can see here that indeed you see a nice tumor control, and if the very same mice are injected only with the engineered immune cells, you see that there is no toxicity. So I think a very, very interesting, and of course in line also with CAR-T, we published in another paper that T-cell persistence is important for a good tumor control. And just in print is another important aspect, because I told you in the beginning that for the gamma 9 delta 2 T cell receptor aspect, we reprogram CD4 and CD8. And the T cell receptor I just showed now only reprograms CD8 positive T cells. And the reason as to why it is because it's a CD8 dependent gamma delta T cell receptor. So we asked the simple question, can we then also reprogram CD4s, and does this improve clinical outcome? And this is what you can see here and soon also online, that if we co-express a CD8 alpha receptor along with this delta 2 negative gamma delta T cell receptor indeed reprograms also CD4 cells. And as you can see here on the right side, it also increases in vivo tumor control in these mice. And last but not least, just some oversight also over activities from different companies which popped up during the last years, where we learned a lot about first-in-man dosing happening from different concepts in this year, and FDA clearance. I think also in this one is now a first-in-man dosing happening. So we are really looking forward to these data. What is take-home message summary? So first of all, we need to better capture and control diversity in numbers before and after transplantation to improve clinical outcomes. We need to restrict diversity in numbers. This can be done with smart Atg dosing, also in an alpha beta T cell depleted setting, because then we need to do it early, or if you prefer alpha beta T cell replete, then think about lymphocyte dependent dosing. And of course, graft engineering is a very interesting way to put it forward, this concept. Also, the choice and dosage of chemotherapy impacts immunological diversity. I showed you the example of fludarabine. Data coming up hopefully soon, and a smooth and post-allo transplantation period is more likely, and in these type of transplantation platforms, and allows early DLIs. But of course, it's all about dosing and in relation to timing. I showed you an interesting twist of the antibody we are using for GMP grade depletion, which might be useful for alpha beta TCR engineering strategies. And of course, the tech concept I showed you, I think is also very interesting now in the clinic tested, and maybe also interesting for delta 2 negative gamma delta TCRs. But be aware it might need some additional help. A lot of people I would like to thank. I think that the first end of COVID, hopefully, a lab meeting we had for all the people and for all the hard work I would like to thank, as well as our collaborators. And at the end of the session, I am more than happy to take any questions from your side. Thank you very much for your attention.
Thanks so much. Alice, looking forward to your talk.
Thank you so much, Leslie, for the kind introduction. To say that I am excited for this meeting, it is really an euphemism. I am really grateful to have you all here. We had an exciting start with the beautiful story of the T-cell depletion by Yair Reisner, and thank you, Jürgen, for this other amazing talk, focusing more on what can be the role of the gamma delta T cells in this setting of alpha beta T cell depletion. What I am going to show you in the next 30 minutes is what is my vision, and I would say the Stanford vision about the alpha beta T cell depletion, and how we really believe that this is a platform which is considered ideal for adaptive immunotherapy. Of course, the reason why we started really to use all these strategies of graft manipulation, of which alpha beta T cell depletion is just one of the most recent, is because we often don't have a donor available. We only have matched sibling donor for 25% of our patients, and despite the presence of over 30 million of unrelated volunteer donors worldwide, there is still a huge proportion of patients who cannot allocate a suitable donor in a suitable frame time. This number is on average 40% when we talk about Caucasian, for example, but it can increase up to 75% if the patient in the need belongs to an ethnical minorities. And that's why really the scientific community in the last decade worked so hard in identifying different strategies that can make haploidentical donors, so donors fully mismatched for an entire haplotype, anyway able to obtain the same outcome that we were able to show having with fully matched related and unrelated donor. And I always like to show this slide that I owe to Mike Pulsifer, who made the editorial to the first paper we published about alpha-beta T-cell depletion back in 2014. Really, he mentioned haplo as the new black. And here in this figure, there are many different strategies that are employed to obtain the same goal, but this meeting is all about alpha-beta T-cell depletion, because I personally believe that this is a strategy which include altogether the most of the advantages of the T-cell depletion, overcoming the main limitations of the T-cell depletion itself. Not that we need so much introduction of what it is alpha-beta T-cell depletion to this audience, but just to remind everybody that when this strategy has been developed, the rationale behind that was really eliminating from the graft the only subset of T cells responsible of developing graft-versus-host disease, those T cells carrying the alpha-beta chain of the TCR receptor, while leaving in the graft, along with a megadose of stem cell, other effector cells, such mature NK cells and gamma delta T cells, that can first being really a first line against the infection in the very early time period, but also having some anti-leukemia properties that can avoid early leukemia recurrence. And in 2018, when I was already here, along with Franco Locatelli and Marco Zecca and Daria Pagliara from the Italian group, we published the results of this multi-center study that we ran retrospectively within the Italian group of pediatric oncohematology. And here the goal was really to evaluate the efficacy of this approach in a multi-center setting. So we collected data from 13 Italian centers performing alpha-beta T-cell depletion, and we compared the outcome amongst three groups: those who receive haploidentical alpha-beta T-cell depleted transplant, and those who receive a match unrelated donor transplant, either 10 out of 10 match or 8 out of 10 or 9 out of 10 mismatch. So main messages coming out from this paper were that when using alpha-beta T-cell depletion, the rate of severe grade 3 to 4 acute GVHD is extremely low. Actually, in this specific analysis, no patients enrolled in the haplo group develop grade 3 to 4 acute GVHD. And the same is for the extensive form of chronic graft-versus-host disease, translating on a non-relapse mortality, which was significantly lower when compared to mismatch unrelated donor transplant. Despite the much lower profile in terms of toxicity, when we look at the leukemia-free survival by donor at 5 years, we saw that the outcome was superimposable in the three groups. Therefore, again, evaluating a long-term composite endpoint, which look at the patients who were 1 year after the transplant alive, disease-free, without developing grade 3 to 4 acute GVHD or chronic graft-versus-host disease, it was clear that if a patient didn't have a 10 out of 10 unrelated donor available, the haplo-identical donor with alpha-beta T-cell depletion was a superior choice in comparison with a mismatched unrelated donor. And just to give you an idea, we believe in the community that this approach is so effective that within the COG, we are going to start a prospective randomized clinical trial in which we will evaluate the haploidentical transplant with either alpha-beta T-cell depletion or post-transplant side against 10 out of 10 unrelated donor. So hopefully we will have soon results that can show if this approach can be considered even superior to a fully matched unrelated donor transplant. But when I moved to Stanford in 2017, we started to use this approach here as well. And I can show you today some of the preliminary results of the transplant we have performed here in 69 patients. In this case, the median age of patients at transplantation was 13 years. We have used as donors mostly the mother or the father, but we also had the chance of using a haploidentical sibling in 25% of those patients. And the median age of the donor for this group was 37 years, with the oldest donor being 55 years old. Here, I can show you a breakdown of the indication at transplantation. 55 patients had a malignancy, while 14 were affected by non-malignant disorders. Within malignancies, the majority of the patients expected as per the age were affected by acute lymphoblastic leukemia, while 33% of them had AML, and then a variety of other disorders, including lymphoma, GMML, and CML. One important characteristic that I would like to draw your attention on is really the high risk group that we are talking about in the specific analysis. About 60% of these patients were in fact in CR2 or greater, and 15% of them had active disease at the time of transplantation because of a refractory disease which was not responsive to the conventional treatment. So a very high risk group of patients in terms of leukemia recurrence. The conditioning regimen was a fully myeloablative conditioning regimen in the majority of the patients, TBI-based for children greater than 3 years and busulfan-based for the youngest one, while just a minority of patients, especially those who received this as a second transplant or who had a high comorbidity index, received a reduced intensity conditioning regimen. And this is just to show you within the non-malignant disorders, the really heterogeneity of the disorders that we had the chance to treat. I will talk to you tomorrow about the outcome of this transplant in Schinke immunoosseous dysplasia patients. We had the chance of treating 2 siblings with an extremely severe IPEX, but also patients with Fanconi anemia, beta thalassemia, Wiskott-Aldrich, and so on. And in this case, of course, the conditioning regimen was tailored upon the underlying disease. As previously shown, neutrophil and platelet engraftment is incredibly fast and robust after alpha-beta T-cell depleted transplant. In our last data, 12 days is the median time to reach the neutrophil engraftment and 15 for the platelet engraftment. The cumulative incidence of grade 2 to 4 acute GvHD for this cohort was 28.8%, with 5 patients developing grade 3 acute GvHD. And here we show the incidence of limited skin-only chronic GvHD, which affected 6 patients and was completely resolved on all patients with a short-term course of immunosuppressive therapy, which in many of these patients was just topical. But what about relapse? So for this specific cohort, the median follow-up is 1 year, so still limited. However, if we look together at the a full cohort, the cumulative incidence of relapse is 27%. But if we limit our analysis to the patients who were transplanted in remission, so patients who did not have an active disease, this number drops down to 16%. So a great result considering, again, the high risk group that we transplanted. And this table, just to show you the graft composition. The graft composition for this transplant is incredibly crucial. And I know many centers who started to use this approach, especially in the US, were or are concerned about the learning curve and the expertise in the laboratory. I can tell you that here at Stanford, we are using 3 facilities for manufacturing. LCGM at Stanford, UCSF facility, and most recently the CTF at Stanford Adult, and I'm extremely grateful for having these 3 teams. And those are numbers coming from the first 2 facilities I mentioned, where the really reproducibility of the graft manipulation has been implemented quickly and in a very robust way. As previously demonstrated, we are able to infuse in our patients a median of 15 million per kg of CD34 with an extensive T-cell depletion, which leave in the graft no more than 40,000 alpha beta T cells per kg as the average, large numbers of both gamma delta T and NK cells. But of course, here today we are talking about what's next in our basket, and specifically what is our strategy at Stanford to really improve the outcomes of our patients now that we have identified a platform that we can define safe and effective. And so when Maria Grazia and I wrote this review in 2018, we really elaborated on what was our vision. Vision for improving the outcome, which means boosting the immune reconstitution, reducing the number of life-threatening viral infection that can really be a problem after a T-cell depleted transplant, but also increasing the leukemia-free survival by promoting immune surveillance. How can we do that? Well, we have the unique opportunity here to have really a great integration between basic science and translational research. And so there are a number of cell products that are in the preclinical development in our laboratories and then get manufactured in our GMP facility and lastly brought to the bedside to the patients. Matt Porteous tomorrow will mention you a preclinical work that we completed together in manufacturing alpha beta T cell CAR from the leftover or discarded alpha beta T cells after the manufacturing. And I will mention you more about this clinical trial that is now ongoing as a phase 1 trial in collaboration with Maria Grazia. We are working though in developing an academic cell product based on gamma delta T cells, and I'll mention briefly in the next slides, as long as we have the luck to have Rosa Bacchetta in our group, who is extensively work along with Maria Grazia on Tregs. This is just to, to show you when I talked about evolution of this alpha beta T cell depletion since I moved to Stanford, what do I mean? Well, my laboratory is really working on trying to develop new cell product that we can ultimately use for the patients. But for doing that, there is a first step which is crucial and is really understanding what we are infusing, characterizing the cells that we are infusing, and how we can modulate their quality to have a most effective transplant, and to ultimately, why not, developing a next generation of alpha beta T cell depletion that can be even more effective. And so this slide is just to tell you where in this graph composition we are working, and we have a fantastic collaboration with Ken Weinberg and his lab that is really pointing out the role not only of the pure stem cell by Stanford definition, but also of the committed progenitors. And so how engineering the graft with common lymphoid progenitors or common myeloid progenitors can improve the outcome afterwards. I cannot show you data on this because as long as they are under embargo because they have been accepted at ASH, but I hope you all can see those data presented in December. And another important part where we are working on, similar to what Jürgen mentioned before, is the clonality of the gamma delta TCR. When we pick a donor, we don't know exactly which characteristic we are looking for from a gamma delta T cell perspective. We don't have a clear understanding on the oligoclonality or polyclonality of the gamma delta T cells and how that can impact on the outcome of our patients. So again, we have a preliminary data that I can't show you today that really can lay the foundation for evaluating the importance of looking at the clonality of the donors to promote graft versus leukemia effect post-transplant. And of course, the alpha beta T cells, even if in this approach are the bad cells, and so the one that we try to eliminate, still have a role in their expansion afterwards, of course, in protecting from the infection in the early phase, but also can have an impact in determining graft versus host disease. And so I'll show you in a few slides how we are trying to understand their role and their impact in developing severe graft versus host disease. So going to the second phase of what is the work we are doing in the lab, here is a visual for the main cell therapies we are working on. You will hear from Robert Lowski the approach that Stanford adults, our big brother, is using. They are using that in integration with the CD34 positive selection, but is really the use of CD8 memory T cells, again, to promote and boost adaptive immune reconstitution post-transplant and hopefully reduce leukemia recurrence. Together with Robert Lowski and his group, we are working in developing a new preclinical product that can really help this DLI in being much more effective and effective specifically for the malignancy we are treating. My passion in terms of T cells lies on gamma delta T cells. One of the main limitations of gamma delta T cells is their low frequency in the peripheral blood, especially for the Wu delta 1 subtype that usually is more residing in epithelial cells. So a work that Priscilla Slepiska is doing in the lab is really engineering these gamma delta T cells with what we call an orthogonal IL-2 receptor, which will be able to expand in vivo safely the gamma delta T cells without bringing all the side effects of the human IL-2. Lastly, but not less importantly, we take advantage of the science of Maria Grazia Roncarolo and Rosa Bacchetta and the development of the T-allotent cell product. This is a product that has been first developed in Italy and then optimized in their manufacturing when they move here at Stanford. And here I'll spend some slides to show you exactly what is the mechanism of the T-allotent cells and how we are using them now in clinic. But first, as I told you, why our strategy is really the one of combining adoptive immunotherapy after the stem cell transplantation. Well, this is because if I have to find a limitation to the alpha beta haplotransplant, it's really in the very early phase after the transplant. This little period of time that you see in red in this line. Because of the extensive alpha beta T cell depletion, we end up in having a window of time which is between the first 45 and 60 days, in which the only CD3 cells circulating are gamma delta T cells. This is, of course, a crucial time for viral infection, but also for leukemia recurrence, especially for patients who did go to transplant with some residual burden of disease. So the goal that we have with the T-allotent cells is really to fill this gap, to give an adaptive transfer of, in this case, CD4 positive T cells, which are donor-derived, so that we can, one, cover this period of time with an adoptive transfer, and two, hopefully trigger a better generation of naive T cells from the donor, along with the adoptively transferred T cells. But so what are these T-allotent cells? This is a cell product which is made by the stimulation of donor CD4 positive T cells, along with dendritic cells coming from the host in the presence of IL-10. During this primary stimulation, one of the specific quality that these cells acquire is called energy. And energy is really in hyporesponsive state that is subsequent to the stimulation with the same alloantigens, the alloantigens coming from the host dendritic cells. This quality is mostly given by the enrichment of this cell product in T regulatory type 1, a specific subset of Treg cells that was discovered many years ago by Maria Grazia, and that is phenotypically characterized by the co-expression of CD49B and LAG3. In addition to their energy, these T alloten cells, as stimulated, become able to suppress primary responses of autologous CD4 and CD8 cells towards the same alloantigens. This property is mainly due to the induction of the TR1 cells, as I meant. However, why it is so important to have this product for really boosting the immune reconstitution? Not only for the ability of promoting tolerance, but because the cells that we infuse remain able to respond to third-party antigens. So even if they have a host alloantigen specific energy and suppressive activity, these cells preserve their ability to respond to third-party antigens, meaning viral antigens or tumor antigens. And so that's where really we believe they can improve the post-transplant immune reconstitution. These figures is coming from a recent paper published by Maria Grazia Group, and specifically from Pauline Chen and Alma Cepica. And it can show you the protocol that has been established at LCGM for manufacturing T cell for clinic use. Is a cell production that overall takes 17 days. And really, in the end, we have two components on this T cell product, which are the TR1 cells and the CD4 effector cells that are infused along with the TR1. Here you can see that in the T alloten cell product, the average enrichment of TR1 is about 10%. And specifically, the T alloten cells, when looking at the cytokine production through qPCR, they maintain the specific cytokine production profile, which is typical of TR1, meaning IL-10 production and IL-22 production with the moderate production of interferon gamma. But also in this paper, in which Alma and Pauline evaluated 51 donor and recipient pair, they can really show that determining the proliferation of T alloten cells and control, specifically when they tested the proliferation through CFSA level cells towards the alloantigen mature dendritic C cells, these cells are unable to proliferate because of their immune suppression, but they still remain able to proliferate when evaluated against third-party mature dendritic cells, for example. And lastly, you can see here an example with the CD4 positive T cells, in which when stimulated with the same host alloantigen manufactured from the host, the suppression is on average 84.6%, while it is not present towards a third-party antigen. We believe that the TR1 component of this product is the one which is really crucial for the success of this product, because really it contains tolerant properties, but also maintain alloantigens properties. And it has been shown here in this evaluation of the transcriptome that the TR1 component of the T alloten cells has a transcriptome completely different from the rest of the cells. And so we are following, specifically Rosa Bacchetta in the lab is following the TCR clonotype of the TR1 component of the T alloten cells that we infuse in the patients to see their persistence post-transplant. Here is the outline of the protocol that is now in clinic. This is a phase 1/1b clinical protocol in which we are using the alpha-beta T-cell depleted transplant as platform after a fully myeloablative conditioning regimen and in the absence of pharmacological immune suppression. We are evaluating 3 escalating doses of T alloten cells, 1 × 10⁵, 3 × 10⁵, and 1 million per kg, infused on day 35 ±7 after transplant. To be eligible to receive the cells, patients need to have evidence of myeloid engraftment and absence of grade 2 or greater acute GVHD requiring over 0.5 milligram per kg of steroids. And for this trial, we have a safety endpoint, which is the primary phase 1 endpoint, of course, identifying the recommended phase 2 dose at 3 different escalating doses. And our efficacy phase 1b endpoint is the evaluation of the immune reconstitution. Specifically, we decided to use as a measure the absolute number of CD4 positive T cells identified as more than 50 CD3, CD4 positive T cells by day 60 after transplant. And just for you to know, in our naked alpha beta cohort, this outcome was reached only in 30% of our patients. So we hope with this approach to improve this immune reconstitution. This is a table to show you the patients that we have enrolled so far. We have enrolled a total of 6 patients and treated 5 of them. We have 3 patients who received this transplant as a second transplant. The donor was a mismatch-related donor, haploidentical in the majority of the case. The graft composition was very consistent for all the patients I mentioned you in terms of CD34 and alpha beta T cell, and the dose of T allotent cells infused for all these patients is 1 × 10⁵ per kg. Again, just to highlight that all these patients are very high risk in terms of relapse, being all in CR2 or greater. The longest follow-up that we have for now is 202 days. So I'm not going to show you data of safety and efficacy because it's too early, but I can tell you that in these patients, we didn't observe any DLT. And more to come about this interesting clinical trial. Last part of my talk is about the role of graft versus host disease in alpha beta T cell depleted haplotransplant. In my eyes, to make this platform really ideal, we need to limit as much as possible the occurrence of severe graft versus host disease. So I'm talking about really grade 3 to 4 acute GVHD. And so Raul Montiel Esparza, who is now a 3rd-year fellow here at Stanford, is leading this effort in trying to understand really the cross-talking between donor immune system and recipient inflammatory milieu. And he's doing that looking at the single-cell cytokine secretion of the alpha-beta T cells residual from the manufacturing and infused into the graft. A single-cell genomics and proteomics evaluation of those T cells as well to identify specific clonotypes that can be responsible of the GVHD. And lastly, looking at the circulating cytokines in the recipient at the time of the stem cell transplant infusion. So the preliminary data that he produced are really about 4 patients that we evaluated with acute graft versus host disease who developed acute graft versus host disease with 3 controls. So patients who had the same exact transplant and conditioning regimen without developing graft versus host disease. Here is a specific evaluation of the methods that we use. This is a technology which is called isoplexis and evaluate a single cell proteomics for both CD4 and CD8 positive T cells. So the cells, the CD4 and CD8 positive T cells have been sorted from the aliquot of the graft, which was infused into the patients. These cells were single labeled with a chip assay. We looked for this specific adaptive immune panel that divide cytokines within 5 functional groups. And here is what we have first shown with this 32-plex antibody array that can measure cytokines in terms of secretion frequency, but also number of cytokines that can be produced by the single CD4 or CD T cells across these 5 functional groups. So T cells have been defined as a polyfunctional when they were secreting at the same time 2 or more cytokines. And you will see here talking about polyfunctional strength index. This is a mathematical evaluation that is defined as the percentage of the polyfunctional cells by the MFI of the protein secreted by the cells themselves. So this plot that you can see on the left is a TSME evaluation that really showed the single cell functional heterogeneity. You can see that highly polyfunctional cells with an effector and stimulatory dominant patterns were present only for the acute GVHD onset graft group. And the single cells polyfunctionality average of the CD4 and CD8 in this histogram on the right show that there is a greater upregulation of the polyfunctionality instead in the non-GVHD recipient compared with the non-GVHD patients. So if we look at the PSI, the single cell polyfunctionality strength index, we can see that the CD4 positive T cell subset is composed mainly of effector, stimulatory and chemoattractive secretion, and CD8 cell subset of effector and chemoattractive secretion. Lastly, you can see this heat map here that indicate how often a combination of protein was observed in a specific given sample. And in these green rectangles, we depict a unique cytokine combination that can really be specific only of those patients who did not develop graft-versus-host disease, specific of the donors of those patients who did not develop GVHD. So what Raoul showed in this first pilot experiment is really how we can identify a specific polyfunctionality in the donors of those patients who develop GVHD and the specific cytokine secretion in those who did not develop GVHD. As another evaluation of the alpha beta TCR is really evaluating the different TCR families in terms of different usage of TCR beta genes. And here you can see an example where he compared a healthy control with 3 haplo patients, 2 who did develop GVHD and 1 who did not. And you can see that these clonotypes are very different, very differently expanded in patients who did develop graft-versus-host disease. On a separate but related note, we are also looking at the role of T-cell alloreactivity and immune checkpoints inspired by the work of Leslie. And this heat map shows differential gene expression for the 3 patients. And you can see that the expression for most of T cells is higher in the haplo recipient versus the matched sibling donor, who is our control, and was also generally higher at the time of the onset of GVHD. Lastly, this is a slide about the cytokine secretion in the plasma of patients at the time of receiving the infusion of the cells, evaluated with a multiplex inflammation panel of 384 cytokines through Olink. This is a differential serum cytokine expression at day 0 for haplotransplant recipient that developed and did not GVHD. And you can see that the cytokines in purple, which are above the line, are cytokines which have been previously demonstrated to be significant for development of graft-versus-host disease. So in summary, the alpha beta T cell depleted haplotransplant really is a platform that now overcome the limited availability of fully matched donors and provide a donor almost for everyone, but also represent the ideal platform for cancer therapy, cancer immunotherapy. How? Well, one is the potential future co-transplantation of additional committed progenitors, CLP, for example, to improve the immune reconstitution, or donor-derived adoptive immunotherapy, as we are doing now in the phase 1b trial with the TLO10 cells. And lastly, we really need to understand, we need to start from the clear understanding of what are the characteristics of the graft that we are using now to modulate and to further tailor in the next generation of the graft manipulation the cells we are infusing. So different directions, TCR of both gamma delta and alpha beta, but also really functionality of the alpha beta T cells that we are infusing right now in terms of leukemia prevention, but also graft versus host disease. And the data that are coming out from my lab are showing that an increased donor T cell polyfunctionality, especially with the T helper 1 dominant functional profile, may be predictive of an increased risk of graft versus host disease. And so the next question, and of course, how are we going to use that that information? Can that really be the start of a new manipulation strategy, or a signal that when using those donors, we need to modulate in vivo into the patient the immune milieu? Before, thank you all for listening to this talk. I really have a number of people to thank for doing this work. As you know, it takes really a village to be able to produce such data. A big thank to the stem cell transplant clinical team, everyone who contributed to this work, but specifically the core clinical people, Rajni Agarwal, Ami Shah, David Shire, and Orly Klein. A special thank to Maria Grazia Roncarolo, Robbie Parkman, Ken Weinberg for their continuous mentorship, my lab, and for this specific work, Giulia Barbarito for the phenotype, and Raul for all the work on graft versus host disease, the incredible clinical trial program, Alicia Boog, Christine Dugal, Gopin Saimi, Natalie Pukesh, Nivedita Conte. I really want to mention them all because they are the people who are running the show. The manufacturing facilities at LCGM, let me mention Catherine Maslop, Kerry Tate, and Niar Bhatia. CTF, Catherine McIntyre, Everett Meyer, and all the team, the UCSF, and specifically Sherman Barabaka, who has been very generous in helping us when we don't have enough space here, and of course, all the agencies which are funding our work. CERM, who lastly funded a prestigious clinical grant, but also many others. And thank you all for your attention.
Thank you so much to both the speakers. That was wonderful. I'm now going to open up the session for questions. Maybe I'll ask the first question. And first, I'll be drummed out of the 7 out of 8 mismatch unrelated donor core if I don't point out that there's actually new data to suggest that we can now make 7 out of 8 transplants actually look very similar to these other historically better alternative donor transplants, so both with PT sign, with abatacept. So just, you know, it's important to think about that. But I'd like to, I have a sort of a general question for both of you, but Alice, just your data on the graft composition and how it impacts you know, events like GVHD was actually really surprising to me. So this is day 0 in the graft that you're looking at, is that right? And how do you sort of think about that? And also given the fact that you've depleted so many of those alpha beta, so what you're looking at is the alpha beta T cells that are left in the graft. Am I right?
You are right.
Yeah.
And you know, what is surprising to me, it is that despite a very homogeneous depletion across the patients, because you saw the number of alpha beta T cells is very, very reproducible. There are some patients, few, but still some patients in which we see some grade 3 to 4 acute GVHD, which really bothers me, to be honest, because, you know, even in the patients with leukemia, you don't want to have a patient that you need to immune suppress because of the GVHD. And so my question is more a question of quality of the T cells that we infuse, more than a question of a quantity of the T cells. And we always thought that using a donor which is older, for example, with an older patient is a risk factor for GVHD. But why is that exactly? Is this really a problem of the donor T cells, or is a combination of the donor T cells that we infuse in a milieu which is already inflamed more than in another patient, and this gets like the nuclear cocktail for really developing GVHD. So really, Raul, what he did right now was analyzing some of the alpha beta T cells residual in the graft after the manipulation. So we are talking about very, very, very small numbers in which just with a single set technology we can analyze. And now we are comparing this data with the T cells of the donor before the manufacturing, because my question is this pattern related to the manufacturing process somehow, or is something that we can identify in the donor to start with, and so can be almost a selection criterion for us. And the other time point that we are looking is the onset of the GVHD. We are analyzing in the same way the alpha beta T cells at the onset of GVHD to understand how this secretion of cytokines changed, if it did, between the infusion into the patients and afterwards, and understanding, one, if we can really pick the donor based on this characteristic, and two, if we just have to pick that donor, is there something in terms of immune modulation in vivo that we can do to reduce the risk of GVHD?
Mm-hmm. Yes, and do you think that the signal may be due to the fact that these patients don't get any post-transplant immunomodulation? Is that?
It is possible, you know, that some alloreactivity is so evident that might require some immune modulation. And I think that this, you know, it might be for really a small proportion of those patients. But since we are using this platform also for non-malignant disorder, I think that in this specific setting, it's really crucial to know whatever we can do to reduce this alloreactivity, to contain this alloreactivity.
Thank you. Maybe I comment on sort of, because what I like is, and we have more differences because you're doing kids, we are doing adults. We are doing also most of the populations is not haplo. We do it just in the unrelated and the SIP setting, because of their we see this great reduced toxicity. And I'm as bothered as you are from this low incidence of really acute GVHD in the early beginning phase. And I have also no answer to that. But I think maybe we should team up, because the experiment you're mentioning, we are also doing in the next months, or as we speak, have already some data. And maybe looking at a bigger cohort, you might have common patterns you can recognize, because I think that's really one of the problems we need to solve.
We'll be happy to do so. I agree that this is one of the problems to solve for sure.
Great. Let me get to some other questions. Dr. Koduri asks, would you recommend depletion cell therapy in AML monosomy 7 patients under the age of 10?
I think that, so I didn't show data breakdown by disease, but definitely the results in AML patients are the same, if not better than in ALL patients. So I would definitely recommend the alpha beta depletion in a mismatch, either related or unrelated donor setting.
And is that true for adults as well?
I leave that to Jürgen.
Yeah, Jürgen.
Short answer, yes. Yeah.
Sounds good. Great. Another question is, Alice, can you tell us if the gamma delta T cell subset composition, VD2 positive versus VD2 negative, may play a role in the transplant outcome and in fighting infections?
This is a very interesting question, and I need to answer with, you know, a preliminary answer. So I didn't show, for limit of timing, the data that Giulia Barbarito is producing about really the composition of the V delta 1 and V delta 2. But I can tell you that there is a very interesting finding. Of course, we know that in the peripheral blood of adult healthy donor, we have more V delta 2 than V delta 1. However, when we look at the immune reconstitution in the patients very early after the transplant, meaning day 30 after transplant, the majority of the gamma delta T cells that we identify are V delta 1 positive, and they have a naive phenotype. So this is a very intriguing finding that we are characterizing with the TCR sequencing. And for sure, there is a role of delta-1 percentage in protecting against the CNV infection, which also translates in a better leukemia-free survival, because we know that when delta-1 are activated upon CNV reactivation, they acquire a better anti-leukemia property. So we didn't have the possibility yet to really look how much the viral infection are distributed based on the graft composition, but this is an information that we will have available soon.
And I think to adding it from the adults, sort of from we made an analysis on CMV reactivations on outcome, and of course it's again limited numbers we have, but it really looks like that in the post-transplant phase, it's way much more important to have a CMV reactivation. So questioning also a lot of strategies which are now coming up where you would like to do CMV prophylaxis from the very beginning of transplantation with letermovir, for example. Is this really something you would like to have? Or shouldn't we just turn it around? Do we just want to propagate, in particular in the setting of a T-cell depleted transplantation where CMV doesn't make the toxicity we know from T-cell replete? Isn't that what we want? And shouldn't we have vaccines who boost sort of this innate immune response? I think that's maybe the way to go, because and that's a challenging question also for Atg. I'm really impressed by all the immune products you will do to the patients or will give to the patients, but I think the true challenge is to come to real life. So how vivid and surviving are your strategies if you go to the big masses? Yeah, because you're treating later a lot of patients, hopefully worldwide, and I think also we need to think about something which is really applicable to a broader patient population.
Absolutely, you are totally right. And indeed, here the goal is to identify what is the best cell product, because I don't know it yet. And I don't think we even have enough, preclinical information to focus all our energies on one cell product itself. Plus, I think that I really believe that the graft composition role for malignancies or non-malignant disorders should be is very different. And then my goal is really to identify two approaches to cell therapies that can be most effective in each group of disease. And in one, we want to avoid any graft versus host disease. In the other one, we want anyway to preserve GVL, so we might need to pay the price of some degree of GvHD. The way is long to go. In my dream, we should be able to evolve the depletion that we have right now with some tweak that can combine what we learned so far about the good and the bad of this depletion. And I hope all these studies will really help us in going into this direction.
Thanks. That was a great sort of in general sort of overview of what the big questions are, right, with this sort of component therapy. Yeah, somebody has asked, as anonymous attendee, is it worth considering a randomized trial between haplo-PT-Cy versus alpha-beta depletion versus 7 out of 8 matched with ABBA? And I think that's kind of gets to that question: is that how do you create the most widely applicable transplant platform that can answer all the needs? I'm not sure we want to discuss that right now, but I would want to maybe end, we're just at the time, is to ask each of you maybe just to tell us all, and maybe Jürgen, starting with you, what do you think is the most important strength, and what do you think is the most sort of compelling weakness that drives your research with the platform you're using? And I'll ask you first, Jürgen, and then ask Atg.
It's a short time to think. I think that the biggest strength is really the very low toxicity you observe, and it's an important platform to study really the molecular mechanisms to further improve the strategy with engineering or vaccination. The major weakness is costs and complexity, because you need to convince people at centers to also do so, and as long you have no data on overall benefit in terms of financial gain, also for the overall society, it will be difficult to convince the community to really change everything to alpha-beta TCL depletion.
Great. Atg, you get the last word here.
I would say that the main strength is represented by the fact that is a very safe approach. the overall, if you look at all the paper published across, US, Europe, the transplant-related mortality is really low, considering that this is a T-cell depleted approach. And as Jürgen was mentioning, it gives you the chance of playing with donor-derived cells, which is really a great advantage. The main weakness to me is really the suboptimal T-cell immune reconstitution and the balance between the improvement of the T-cell immune reconstitution and the graft versus host disease. We already improved so much from the immune reconstitution that we were seeing with the CD34 positive selection, but still now we need to do the next step up to really make this perfect.
Great. Thanks so much, you both. We're out of time. I think, Alicia, correct me if I'm wrong, but there's a break now until 2 p.m. Eastern and 11 a.m. Pacific time. Is that correct?
Is that correct? We are all have the chance to have a little break. I thank you so much, Leslie, for moderating this session. That was wonderful. Thank you, Jürgen, for participating, and I'll see everybody back at 11 a.m. Pacific time. Thank you.
Thanks, everyone. Bye.
Bye.